Next up is Daniel Singlearm, and he represents Keystone Bio. Keystone Bio is a remarkable product looking to address a bacterial infection issue that I didn't even know about, but many of you probably do, but I guarantee you don't know how serious it is, but they're solving that problem for us. So Dan, take it away for us.
Excellent. Thanks for the introduction, and good to see you all. So over the last 15 years, I have been, as I tell Ed periodically, I eat, drink, and breathe oral systemic health. I noticed back in the 2000s, I was a practicing wet-fingered dentist, and all of my patients that were in their 80s and 90s that were living very full lives all had the gums of a 16-year-old. And, there's really nothing you can have in your 80s and 90s that's a 16-year-old. So they had extraordinarily healthy mouths. And the ones that didn't were moving on to disease. So the outcome of that was, this is not about me, but I took a preceptorship, first dentist to take a preceptorship in heart attack and stroke prevention. The wanted need there was how to get the driving forces of the mouth off the table for everybody's patients, for physicians. And the outcome formed American Academy for Oral Systemic Health, progressed through the early 2010s. Probably about 2014, it became very evident that there was one specific keystone pathogen that was driving this problem. I'm the CEO of Keystone Bio. We're an early clinical stage biopharmaceutical company targeting that major driver of disease, the keystone pathogen Porphyromonas gingivalis. PG, I'll call it PG from now on. PG is a killer. It has the ability to cause numerous disease states. Keystone Bio has discovered why, and I'll go through some of that in a little bit. There's a huge problem. The consequences. Keystone Bio has the only biotherapeutic that's been shown to get rid of PG, and there's a huge opportunity here for all of us to improve millions of lives. Porphyromonas gingivalis in the mouth drives morbidity and death through systemic inflammatory disease. We have identified, while PG lives in the mouth, and there's 30 to 40% of adults that are positive for PG, we've identified toxic proteins that are released into the blood from PG, transported to various end organs and initiating inflammatory disease. It's been shown to elevate each and every one of the typical inflammatory biomarkers that we all get tested for. It is involved with most inflammatory pathways, and it's been found to release these toxic vesicles. I'm going to show my pointer. This is an oral biofilm, and oral biofilm is becoming a target of a lot of attention in research. There's over 10,000 papers on PG, and these are actual Porphyromonas gingivalis bacteria in the outer wall of the biofilm. These little blebs really play into why Keystone Bio has an innovative and one-of-a-kind way to get rid of those. PG has been shown to evade our innate and acquired host defenses, and there are no treatments currently to get rid of that. And it's been a proven contributor to vascular inflammation, cardiometabolic diseases, and neurodegenerative diseases, including Alzheimer's. So as I mentioned, there are over 10,000 research papers on PG. There's a lot of research showing so in this particular study, it reduced, by reducing PG, it was equal to or better than statins. PG also has been shown to be a known driver of Alzheimer's disease. Our research shows in the Journal of Alzheimer's a large amount of these toxic vesicles in the brain. As late as just 2 months ago, the GAIN trial released results showing that targeting Porphyromonas gingivalis in the mouth substantially slowed cognitive decline by as much as 50%. Nothing else has been shown to do that. We can now identify through oral testing and treat one of the best known major causes of sporadic Alzheimer's disease at its source. So the solution. Keystone Bio has the only therapeutic, it's precision monoclonal antibody that binds to those blebs that I was showing before. This is one of the bacteria, and all these little blebs are the toxic vesicles that are released throughout the body. Here we show in these slides our monoclonal attaching to those, basically killing. This monoclonal has been shown in human trials safe and effective to get rid of Porphyromonas gingivalis. So Keystone Bio has partnered with over 40 groups around the world. We have an Australian subsidiary. We're a full-blown clinical stage biopharmaceutical company. We have a mapped out strategy for our Series B. We've been through Series A. We have CGMP manufacturing of the product. We have tox studies finalized. We have done groundbreaking research and development. We have diagnostics in the pipeline. We also have an eventual vaccine potential going forward. Our Series B will take us through phase 2. Because of the large market that we're in, we see there's a good opportunity for orphan drug or status. There's also good opportunity for fast track and for compassionate use. We have put together a team that includes experts in their field, and we are all passionate about this subject. We've come from different areas of our particular fields: cardiovascular disease, biologics, and vaccinology, myself with oral systemic, and we are set to address all of these problems.
Great. Thank you so much, Dan, for your presentation and for joining us. Super impressive in terms of the research that you've done. So it seems that, when applied topically in the mouth, it substantially slowed cognitive decline and the progression of Alzheimer's disease. Is that right? And is that all by treating P. gingivalis, nothing else? So
that study, the GAIN trial, was performed by another group. They were able to show they lowered the load of PG by 70%, 70, and they had good results with those that had, exceptional results with those that had PG positive in their mouth. Our paper shows we would suspect it would never stop the flow of the toxins, but at least they may have had a pretty good result because they slowed it. Their particular asset was just put on FDA hold. It's a small molecule that's taken a certain number of times a day with a large dose. We have something that's very simply applied. Our particular treatment, it's a unique approach. We have a monoclonal antibody that binds to all of the energy sources of this bacteria. Those also are intricate with the toxins that are secreted into the blood that are transported to other sites in the body. So we have a monoclonal that binds to that, virtually killing the bacteria, already shown up to 9 to 12 months in humans. To put it into perspective, a dental cleaning never gets rid of the bacteria, just slightly disrupts the biofilm. The biofilm completely reestablishes with PG in about 7 days. Periodontal treatment, the same, never eliminates it. It returns within 30 days. So there is never a stoppage of that flow. We have a monoclonal that's painted on the gums. We envision this. It's a very safe, simple placement done by a dental professional. We assume they will be doing this every 6 months at your dental visit, painted on. I don't want to say it's painted on like fluoride, but it'll be rather simple. And we have diagnostics in place. So we will also be monitoring screening and monitoring treatment and diagnosing. There's also some real unique patterns we're seeing where, how could something cause so many diseases? Well, it's an extremophile. It's actually an apex predator we see. Nothing like anything we've ever seen. But then there are different, the host has different presentations with genotypes and with lifestyles that cause a little different approach to how the bacteria takes advantage of them. But this is an underlying driver of many, many diseases.
Dan, another question for you. Why doesn't the body have a natural defense system against the bacteria? Can you tell us a little bit more about why this bacterial strain is so evasive, why this has to be dealt with through a therapeutic, and the body can't eradicate this itself?
So it is the most extraordinary biological or evolutionary anomaly. This thing is like nothing we've ever seen. If you took H. pylori, this is H. pylori on steroids. It has its defense systems actually manipulate our immune system, both innate and adaptive. And it actually uses its own approach to that to feed off of some of the byproducts of our defenses. My first initiation into looking at PG was in 2010. There was a paper showing that it simply transported or changed our immune system from a destroyer to a carrier. So it has all kinds of, it has 151 virulent proteins to it. It has LPS, real intricate systems that explain why it's so readily able to do that. My guess is because it's a strange combination of an oral bacteria that has a systemic impact. So it's kind of grown up in the oral cavity, and the oral cavity is an environment where there's a lot of manipulation with inflammation. The same process to a certain extent that occurs in the mouth occurs in the body. The body isn't able to be so resilient. We disrupt biofilm, as practitioners in dentistry all the time. We go in for our cleanings and our scalings, and the body gets back to a healthy state. Well, we don't have that ability when it's shooting out these toxins into the body.
Thank you so much, Doctor. And thank you also, Dr. Krell. I have one question from the crowd that I'm going to throw in with, and then we actually have to move on to our next speaker. The question is basically the chlorhexidine mouthwash claims to kill P. gingivalis. How does this technology differentiate itself from that mouthwash and from other potential antibiotic treatments or the GAIN treatment that you mentioned, the GAIN study from earlier?
So, and I'm glad you asked that question. So Chlorhex, in the UK, Chlorhex is used over the counter, and it's used a lot. And they have done numerous studies, not to be picking on the makers of Chlorhex, but Chlorhex it's not supposed to be used as a long-term treatment. It disrupts the nitric oxide pathway by killing a lot of the good bacteria in the mouth. It is not a precision approach. Ours is a precision approach. There actually are studies showing that chronic use of chlorhexidine rinses do increase risk for myocardial infarctions. So we take an approach of a precision, the bad actor. We feel that allows the healthy oral biome to fill in the place.
And then compare that to other antibiotic treatments or to other potential treatments that might be out there?
No antibiotic has been shown effective, and we watch this landscape well. There's attempts on goal for this, but the biofilm has been very productive. If I'm suspecting that, and unfortunately the photo I was trying to show shows PG in the wall, and then how we attach to it and take that out. That is pretty much a foundational portion of the oral biofilm. And we have on our team Garth Ehrlich, who co-developed the biofilm hypothesis. He can speak to that much better than I can, but we have researched the intricacies of oral biofilm beyond compare, and this is a keystone actor in that system.
Excellent. Thank you. And there's lots of other questions we didn't get to, including like who would qualify for treatment and their pathways to market. I encourage everyone to join Keystone Bio in the networking session afterwards to answer those questions and any other that might have come up.